Welcome friends to a new session on genomics and proteomics. In this session, we will discuss gene prediction. Basics of gene prediction. In computational biology, gene finding or gene prediction refers to the process of identifying the regions of genomic DNA that encode genes. This includes protein coding genes as well as RNA genes, but may also include prediction of other functional elements such as the regulatory regions. For example, archaeal and bacterial genes typically comprise uninterrupted stretches of DNA between a start codon, usually ATG, but in a minority of genes GTG, TTG or CTG, and a stop codon TAA, TGA or TAG. Exceptions to this rule involve important but rare mechanisms such as programmed frame shifts. There seems to be no strict limits on the length of the genes. Indeed, the gene RPMJ encoding the ribosomal protein L36 is only 111 base pair long in most bacteria, whereas the gene for Bacillus subtilis polyketide synthase PKSK is 13,343 base pairs long. mRNAs shorter than 30 codons are poorly translated. So, protein coding genes in prokaryotes are usually at least 100 bases in length. In prokaryotic genome sequencing projects, open reading frames ORFs shorter than 100 bases are rarely taken into consideration, which does not seem to result in substantial underprediction. In contrast, in multicellular eukaryotes, most genes are interrupted by introns. The mean length of an exon is approximately 50 codons, but some exons are much shorter. Many of the introns are extremely long, resulting in genes occupying up to several megabases of genomic DNA. This makes prediction of eukaryotic genes a far more complex problem than prediction of prokaryotic genes. Two basic approaches. Two approaches have been adopted for the problems associated with gene prediction: similarity based and ab initio techniques. The information gleaned from these methods is then combined via a variety of algorithms, including dynamic programming or the hidden Markov model, and then used for gene prediction from the genomic sequences. The amount of available genome data is increasing exponentially with the completion of a host of genome sequencing projects. Computational gene recognition programs are critical with regard to the automatic annotation of uncharacterized DNA sequences. Therefore, the perfection of computational techniques for the location of protein coding regions in uncharacterized genomic DNA sequences constitutes a central issue in the field of bioinformatics. It is then crucial to have reliable tools for the automatic annotation of any given DNA sequence of an organism in which the genes and the other functions are not currently known, including the number and location of genes, the location of exons and introns in eukaryotes, and their exact boundaries. Thus, along with standard molecular methods, a host of new techniques for the determination of distinctive features of protein coding regions have been proposed over the previous 2 decades. Intrinsic and extrinsic classes. These techniques can be divided into 2 classes: intrinsic and extrinsic. Intrinsic or ab initio methods deal with DNA sequences and extract information regarding gene locations using statistical patterns outside and inside of gene regions, as well as those patterns typical of gene boundaries. Extrinsic or similarity based methods utilize information derived from similarity search procedures using the putative proteins derived from a list of open reading frames as queries. Combinations of both methods have been determined to perform efficiently in terms of gene annotation in large scale genome sequencing projects. Gene discovery in eukaryotic organisms is more difficult than in prokaryotic genomes due to their low gene density, which is the result of the presence of introns in the coding regions. In order to help and speed up the time consuming duration process and improve available annotations, computational methods able to provide prioritized lists of predicted annotations are paramount. Ab initio gene predictions. Complete genomic sequences are becoming more and more abundant. Given a new genome, one of the first and most important tasks is determining the structure of its protein coding genes. Ab initio gene prediction plays a critical role because it produces gene structures quickly, inexpensively and reliably. The accuracy of a gene finder therefore depends on many factors. Chief among these is proper training. Training a gene finder can be a laborious task. In the past, genome projects moved rather slowly and the trickle of data meant that parameters could be estimated before the genome was complete. Today, sequencing and assembly are so rapid that genomes can appear well in advance of proper training material. This leaves some new genome annotation projects without an appropriate gene finder. In silico methods. Finding protein coding genes in eukaryotic genomic sequences with in silico methods remains an important challenge in computational genomics. The existing methods fall into two groups with respect to the data they utilize. The first group consists of ab initio programs which use only the query genomic sequence as input. Examples are the programs GenScan, Augustus, HMMGene, which are HMM based, and GeneID. The second group of gene finding methods, the extrinsic methods, comprises all programs which use data other than the query genomic sequence. Some extrinsic methods use genomic sequences from other species. Some of the best available ab initio gene prediction programs have been extended and have become more accurate by exploiting additional extrinsic data sources such as the protein or EST sequences. GenScan, for example, has an extension called Genome Scan that uses BLAST alignments with protein sequences. HMMGene has an extension that integrates into the HMM information from BLAST alignments of the query sequence with cDNA, EST and protein sequences. Another program which uses extrinsic evidence is GeneWise. It aligns a protein sequence to a genomic sequence. Further, Exon Hunter allows, like the method presented here, to integrate hints from both cross species comparison and protein and EST databases. Besides the two groups of programs described above, there are also programs like Combiner that combine the predictions of several individual gene finders. The above mentioned programs use statistical models of substructures of genes such as splice sites, exons or introns to derive evidences about a gene structure. A major challenge in computational gene prediction is therefore to design a good statistical model that are able to deal with different types of information from heterogeneous sources. When new genomic sequence emerges, it must be annotated with something, and frequently this is a gene finder for a completely different genome. A particularly good example of this is GenScan, which was trained primarily for the human genome, but has been used to annotate genes in worms, flies, fish, fungi, amphioxus and others. Since sequence features such as codon bias and splicing signals vary from organism to organism, it is expected that gene finders may not perform optimally in a foreign genome. While the practice of annotating a genome with a foreign gene finder is commonplace, its consequences are not widely understood. In order to improve gene prediction, extrinsic evidence on the gene structure can be collected from various sources of information such as the genome-genome comparisons and EST and protein alignments. However, such evidence is often incomplete and usually uncertain. The extrinsic evidence is usually not sufficient to recover the complete gene structure of all genes completely, and the available evidence is often unreliable. Therefore, extrinsic evidence is most valuable when it is balanced with sequence intrinsic evidence. Probabilistic logic programming also offers a powerful way to describe and evaluate structured statistical models.